Hi everyone. In this lecture, we're going to talk about microbial metabolism. In essence, we're going to talk about how bacteria get their energy, what they use for energy, such as sugars, carbohydrates, and how they break it down to get their energy, which for organisms, that's in the form of the molecule called ATP. So we're going to talk about it. We're going to talk about respiration, which can include oxygen, aerobic respiration, or no oxygen, anaerobic respiration, and another type of metabolism called fermentation. So we will talk about all about that, and then we will go through the specific steps that happen and everything. So we're going to talk about how microbes get ATP. So the lecture outline for today is going to be about catabolism and anabolism. Metabolism basically means all chemical reactions that a cell does. So how do you break down molecules to get energy, and then how do you use energy to build molecules? That's what catabolism and anabolism are. I'm going to cover that. We're going to look into ATP a little bit further about why it is the energy currency of the cell. I'm going to talk about enzymes and what they are briefly once again, and then we're going to talk about why cells break down. That's what catabolize mean, or catabolism, sugar, glucose for energy. So how they exactly break it down, and then these are the cycles that they go through to break down glucose. We'll talk about those. And then we're going to talk about, can you also break down proteins, other carbohydrates, and lipids? And then anabolism is how you make components of a cell. So that requires energy. And then finally, we're going to end up the lecture talking about amphibolic pathways. Amphibolic is just a fancy word for saying pathways that have both catabolism and anabolism in them. So to start off the lecture, all cells in the world, whether you're a prokaryotic cell or a eukaryotic cells, need to do two things. They need to make new cell parts. So your cells and bacterial cells and yeast cells and mold cells always have to make new cell parts. And they also have to come up with energy so that they can make new cell parts. So you need to make energy so that you could use the energy to create different cellular components. And the combination of all these reactions, whether it's making energy or using energy to create things, is called metabolism. So the definition of metabolism is the sum of all reactions in a cell. And so microbial metabolism, it's important to study because when we understand microbial metabolism, we can do so many things in the biotech industry. So you can make biofuels, new ways of getting energy out of microbes. We can use it to produce food. So when we understand microbial metabolism, we can get microbes to make a lactic acid that we use in yogurt and cheeses. You can use alcohol and various food products. We can use it as a model to study in labs, so it's study metabolism in humans. And also in the future, we can look at microbial metabolism to create new antibiotics. So maybe a bacterial cell makes a certain component of its metabolism that we can target when we create antibiotics. So it is very important to understand the metabolism of cells so that we can stop them from causing infections, as one example. But these are all the human implications. And again, metabolism is the sum of all chemical reactions. So it includes anabolism and catabolism. Anabolism means building molecules, so building cell components. Catabolism is breaking them down to release energy, which is ATP. So metabolism, again, is separated into two parts, catabolism and anabolism. Catabolism is when we degrade or break down large molecules to release energy. It's the same reason why humans eat. So the reason we eat as organisms is so that we can get energy. Without food, we would die because we need that energy to fuel all our cellular reactions. And so bacteria and other microbes also need to do the same thing. So breaking down or degrading components, that's called catabolism. I want everyone to know that. And when we break down molecules, we release energy, and the reaction involved is hydrolysis. Remember, hydrolysis is to break components. An example of a catabolism reaction is the catabolism of glucose. So when we grow bacteria in lab, we add sugar in the medium. Same reason why humans eat carbohydrates or sugar, so that we could get energy. They're exergonic reactions, meaning they release energy in the form of ATP. And this ATP is important because we need that energy so that we could build different components of cells. So the breaking down part of metabolism is called catabolism. The building up is called anabolism. Anabolism are biosynthesis pathways, meaning that they're pathways that use energy when you want to build cell components such as a cell membrane and proteins and the cell wall. It uses energy, so it's an endergonic reaction. An example of a reaction that is anabolic is photosynthesis. So this is where we build macromolecules. Remember in the previous lecture we learned about the 4 macromolecules. So there are carbohydrates, proteins, lipids, and nucleic acids. When we break these molecules down, that's called catabolism. We break them down for energy, for ATP. When we want to build them, we use the ATP. That's called anabolism. And the combination of both is metabolism. So it's both of these reactions happening. Okay, and then just remember that the take-home message from this is that anabolism uses energy, catabolism creates energy. And both of these processes are linked because they're linked by ATP. So you need to eat, you need to provide organisms food such as bacteria and lab so that you can make ATP, and then you need to use ATP to build macromolecules for the cell. So ATP links the 2 processes. And again, the endergonic process is anabolism. It uses up energy. The process that is exergonic, that releases energy, is catabolism. So this is the main take-home message from these slides. Okay, now we're going to discuss ATP a little bit further. I know I covered it before in the previous lecture. ATP is the energy molecule of the cell, and the reason why it's the energy molecule of the cell is because of the structure of ATP. ATP stands for adenosine triphosphate. When we look at ATP, there's an adenine group, which is a nitrogenous base, meaning you see nitrogen in it. There's a ribose sugar. I know this is a sugar because it ends in OSE. All sugars or carbohydrates end in OSE. And then there are 3 phosphate groups. These 3 phosphate groups are very negatively charged. So P and O are both negatively charged, and when we put all these phosphate groups together, it's a lot of negative charge. And remember from chemistry, negative charges don't want to be next to each other. So there's a lot of potential energy there. So these bonds are unstable, and they can be easily broken by hydrolysis to release energy. So that's why ATP has a lot of potential energy, because of these 3 phosphate groups. And so if your cell needs to use energy to do something, it will take ATP, and by releasing one of the phosphate groups, you get energy. So ATP can be converted to ADP, which is adenosine diphosphate, and then ADP you can extract even more energy from it, and that will hydrolyze or get broken down into AMP, adenosine monophosphate. But the highest energy molecule is adenosine triphosphate, more than ADP and AMP. And it's because of these 3 phosphate groups that are highly negatively charged. Now we're going to talk about enzymes. So before I get into metabolism, I just wanted to talk about what ATP is, what enzymes are. So cells, in order to do metabolism, to break down things and also to create things, they need ATP, but they also need enzymes. Enzymes are proteins. So in the macromolecule world, if I ask you what is the one function of protein is that they're enzymes. Enzymes are proteins that speed up chemical reactions within cells. That's the definition of an enzyme. It's something that speeds up a chemical reaction. Now, a chemical reaction will still happen in a cell regardless if there's an enzyme or not, but it will be so slow without an enzyme. It can be years long for it to happen. That's why we need enzymes. They're crucial for metabolism. So again, they're proteins that speed up chemical reactions. And enzymes usually end with ASE suffix. So lactase is an enzyme. Hydrolase is an enzyme. Anything that ends with ASE is an enzyme. And we say that they're biological catalysts. So they speed the conversion of substrate into product without being altered. And they increase the reaction rate of any chemical reaction. They're highly specific. When I say highly specific, I mean that they only react on their specific substrate. Substrate is what an enzyme reacts on. So the lactase enzyme, and I know it's an enzyme because it ends in ASE, reacts on the substrate lactose. So lipase reacts on the substrate lipid. So that's what I mean by they're very specific. They will only react with their specific substrate to create the products. And enzymes are important in metabolism because without them, chemical reactions would take forever to happen. So again, they're proteins that speed up chemical reactions. I want you to know these details about them. They're highly specific. Again, they're specific to only the reaction they catalyze. So lactase will only act on lactose. They're unchanged by the reaction. So enzymes, what they do is basically they bring all the components together that need to do the reaction, and then the reaction happens. So the enzyme is unchanged in the whole reaction. It's not degraded, nothing happens to it. The enzyme will combine with the substrate to form an enzyme substrate complex, and then the products will be formed. So for example, lactase, this enzyme, will break down lactose. So lactose is the substrate into two sugars, glucose and galactose. So most enzymes end in ASC. So that's what I want you to know about enzymes. And again, they're unchanged in the reaction. So the enzyme that breaks down lactose will break down lactose and then leave the reaction unchanged so that it can break down more lactose. There are many different things that can affect enzyme activity because enzymes are proteins, so they can be denatured. Proteins can be denatured, meaning their structure can be changed, and things that can affect the structure of proteins or enzymes are temperature. So temperature that's too high can make enzymes non-functional. That's why fevers are bad. So fevers, they are good in the sense that they help you fight off microbes, and we'll talk about that later. But the reason why a prolonged fever that's very high can be very bad because eventually that fever will start denaturing your enzymes, meaning making them non-functional. Enzymes can also be affected by pH. So if pH is too low, if you add an acid to an organism or a base, you can kill the organism because you affect the enzymes. They can be affected by salt. That's also why we use salt to kill bacteria, because you affect their enzymes. Regulators, so regulators are things that, for example, activate enzymes. So an activator will cause an enzyme to be activated, and then inhibitors can affect enzymes in that they inhibit or stop enzymes from functioning. So cells require so many different enzymes to carry out metabolism, and we're going to talk about these enzymes in metabolism. So just keep in mind that whenever I say a word that ends in ASE, it's an enzyme. And so now we're ready to talk about energy production in a cell, and that is glucose metabolism. So energy, ATP, is produced in a cell when a cell breaks down a component of a macromolecule like food. So we're going to focus on catabolism of carbohydrates. Carbohydrates are sugars. We're going to talk about how cells gain energy from breaking down glucose. Or for example, I like to tell my students that imagine why you gain energy when you eat a Snickers bar. A Snickers bar is like all glucose. So why do you get ATP? So we're going to talk about the process. And so this breakdown of sugars or carbohydrates to get energy or ATP is called cellular metabolism. So anytime you see someone saying we're learning about cell metabolism, they're basically teaching you how we're going to break a component such as carbohydrate and get energy, ATP. So that's what we're going to talk about. And the cell respiration pathways we're going to talk about are just regular cell respiration, which can be aerobic or anaerobic. We're going to discuss that. And then another metabolism called fermentation. So glucose metabolism. So ATP can be produced from food, from carbohydrates, by either respiration or fermentation. Respiration gives a higher total ATP than fermentation always. So if a cell had the choice whether they're going to do respiration or fermentation to get ATP or energy, they will always use respiration because respiration will give you more ATP. In this image right here, I'm going to break it down even further. I just wanted to give a big overview. Again, the main message here is we're starting out with glucose and we're seeing how do we break it apart all the way until we get ATP, which is energy that the cell needs. So we're going to talk about 3 different types of glucose metabolism, or how we can break down glucose in 3 different ways. Depending on the microbe, the cell type, they will use different types of metabolism or respiration. One type of metabolic process called aerobic cell respiration, this is what humans do. So aerobic respiration or aerobic cell respiration produces 38 ATP total. We'll talk about the details in this table later on to summarize it, but the take-home message I want everyone to know is that there's 3 types of metabolism: aerobic respiration, anaerobic respiration, and fermentation. The one that produces the most ATP by breaking down glucose is aerobic cell respiration. So if a cell had a choice to do any of these, it would always choose aerobic respiration because you get the most total ATP. Anaerobic respiration gives you less than 38 ATP, but more than 2, depending on what you metabolize or what you break down. And then fermentation gives you the lowest number of ATP, and it's only 2 ATP. So we're going to talk about these 3 different types of metabolism. Again, they're aerobic cell respiration, anaerobic cell respiration, and fermentation. And the one that produces the most ATP is aerobic respiration. The one that produces the least amount of ATP is fermentation. And cells always would like to produce the most amount of energy, the most amount of ATP. So we are going to cover right now, we're going to focus on the details of respiration. We're going to talk about both aerobic and anaerobic respiration, and then we'll talk about fermentation. So we'll start. So the energy source, whether you're doing aerobic respiration, anaerobic respiration, or fermentation, that we're going to talk about is glucose. So we're going to say that when you start out with glucose, when you have cells growing in lab like bacterial cells, or a human eats sugar, glucose, how is it broken down? And cells usually use glucose because it's high in energy. Glucose, the formula for it is C6H12O6. So we break down glucose into simpler molecules by respiration or fermentation. Both of these pathways involve coenzymes called NAD+ or FAD+. We're going to talk about these CSAR electron carriers. Both have a terminal electron acceptor. I will talk about it later, and both pathways create ATP. But if I was in class, I would ask everyone again, which one creates more ATP? Respiration or fermentation? I want everyone to know it's respiration, and specifically aerobic respiration. So now the only pathway I'm going to talk about moving forward is aerobic cell respiration, and then we're going to change. So aerobic cell respiration, aerobic means oxygen. So aerobic cell respiration requires oxygen. So you have to be in an environment where there's oxygen, and these cells can utilize oxygen as a final electron acceptor. We'll talk about what final electron acceptor means, but for now I just want you to know that aerobic cell respiration uses oxygen. It produces the most amount of ATP. It produces 38 ATP in bacteria and 36 ATP in eukaryotes, for example, in humans. So when bacteria, if you have bacterial cells growing in lab, if you give them a little glucose sugar, they will do, if they can, they will do aerobic cell respiration, and they will produce 38 ATP. So the steps of aerobic cell respiration that we're going to cover, there's 3 steps. So we're going to say, how do you even get 38 ATP? The cells go through 3 steps. The first step is glycolysis. Lyse means breakdown. Gly comes from glucose. So remember, we're starting off with glucose. So the first step in aerobic cell respiration is glycolysis, breaking glucose. Then there's the Krebs cycle. So this right here is the Krebs cycle. So we start with glycolysis in aerobic cell respiration. Then we have a transition step, so you make something called acetyl-CoA. Then you have a cycle called the Krebs cycle, and then finally the 3rd step is the electron transport chain. So again, the 3 steps in aerobic cell respiration are glycolysis, then the transition and Krebs cycle step, and then finally the electron transport chain. Now we're going to talk about glycolysis. Glycolysis is when you take glucose and you break it down into 2 pyruvate molecules. So when we oxidize glucose, remember from chemistry, oxidize means you lose electrons. So when you take glucose and you break it down into pyruvates. Pyruvate are 3-carbon sugars, and you produce a net of 2 ATP. And then this happens, glycolysis, if I were drawing a cell, glycolysis always happens in the cytoplasm of a cell, where glucose enters the cell and then it's broken down into 2 pyruvate molecules. It happens in the cytoplasm. And what you get produced after glycolysis is you get 2 pyruvate molecules produced, which are 3-carbon sugars. And this makes sense because glucose is a 6-carbon sugar. So if I take a 6-carbon sugar and I break it in half, I get 2 3-carbon sugars. You get a net of 2 ATP, and you get 2 molecules called NADH. NADH is an electron carrier. This will be important. The take-home message I want everyone to take from this is that the first step in aerobic cell respiration is glycolysis. Glycolysis produces 2 ATP, it produces 2 pyruvate molecules, and it produces NADH. Now, from now on, whenever I say NADH or FADH2, I want you to know that they all go to the electron transport chain. We'll talk about that later, but so far we've produced 2 ATP. So glycolysis always produces 2 ATP. Now, in the beginning, I told you that aerobic cell respiration produces 38 ATP total. So we still have a lot more ATP to create. The next step after glycolysis is the transition step. So we take our pyruvate that we created, our 2 pyruvate, and they get converted to acetyl-CoA, and then acetyl-CoA enters the Krebs cycle. So step 2 after glycolysis, which is step 1, is the transition step. So each pyruvate gets converted to a molecule called acetyl-CoA. Acetyl-CoA enters the Krebs cycle. And what I want everyone to know is that the Krebs cycle, so this part right here, in prokaryotes, it happens in the cytoplasm, and in eukaryotes, it happens in the mitochondria. So for all prokaryotes, for all bacteria, glycolysis and the Krebs cycle happen in the cytoplasm. And we start with 2 pyruvates. They get converted to acetyl-CoA. Acetyl-CoA enters the Krebs cycle. And then after this step, the transition step and the Krebs cycle is also called the tricarboxylic acid cycle. Krebs is a scientist who discovered it. So they're the same thing. So when you hear me say TCA cycle or Krebs cycle, it's the same thing. After this step, we get 6 carbon dioxide produced. By the way, this is why we exhale CO2. So it's a byproduct of metabolism. 2 ATP, and then you produce 8 NADH and 2 FADH2. And as we get to the end, all of this will make more sense. So as a reminder, in glycolysis, we produce 2 ATP and NADH and pyruvate. In the transition step, we produce 2 ATP total, 6 CO2, and 8 NADH and 2 FADH2. Now, glycolysis produced 2 ATP, the Krebs cycle produced 2 ATP, but we still have 34 ATP to produce because the whole glycolysis, Krebs cycle, and electron transport chain produced 38 ATP total. So the final step in aerobic cell respiration is the electron transport chain. The electron transport chain is a series of electron carriers in the cell membrane of organisms. So in prokaryotes, the electron transport chain happens in the plasma membrane, and in eukaryotes, it happens in the mitochondria. So to summarize a little, glycolysis happens in the cytoplasm for both prokaryotes and eukaryotes. The next step, which is the transition step and the Krebs cycle, for prokaryotes, it also happens in the cytoplasm, but for eukaryotes, it happens in the mitochondria. And then the electron transport chain happens in the cell membrane for prokaryotes, and for eukaryotes, it happens in the mitochondrial membrane. So keep that in mind. After the electron transport chain, we produce 34 ATP by a process called chemiosmosis, and we also produce water if oxygen is the final electron acceptor, as seen in aerobic cell respiration. So the 3 steps of aerobic cell respiration are glycolysis, Krebs transition and Krebs cycle, and electron transport chain. The process that produces the most ATP is the electron transport chain because glycolysis produces 2 ATP, the Krebs cycle produces 2 ATP, and the electron transport chain produces 34 ATP. The reason why you get 34 ATP in the electron transport chain is these molecules I mentioned before called NADH and FADH2 all go to the membrane, the cell membrane in prokaryotes and the mitochondrial membrane in eukaryotes, and they shuffle their electrons, creating this gradient, which I will talk about. And as the electrons go down their concentration gradient, you get ATP produced. So this is how the electron transport chain works. So we call this chemiosmosis. You create a proton gradient. And so again, everyone realizes this happens in the cell membrane of prokaryotes, of bacteria, and then in the mitochondria of eukaryotes. So NADH and FADH2, they end up going all to the electron transport chain. What they do is they give up their electrons, and by shuffling their electrons in this chain, it creates a gradient of protons. So there's more protons on one side than there are on another side, and the protons want to balance out this gradient. So this proton gradient, which is called the electrochemical gradient, will create this kind of electricity circuit where you have more positive charges on one side than another side. So these protons will want to get back in the cell, and they'll go through a membrane protein called ATP synthase. And every time the protons get back through, they create an ATP. So all the NADH and FADH2, which we call electron carriers, will go here. The flow of electrons will create this gradient, this proton pump, and these protons will want to get back in the cell. And every time they go through ATP synthase, they cause that protein to create an ATP. So this is how this process is done. This is a very short summary of how it's done. It's a lot more detailed than that. But I want you to know that in aerobic cell respiration, oxygen is the final electron acceptor. In anaerobic cell respiration, it's anything that's not oxygen. It's an inorganic molecule. So that's the take-home message of the electron transport chain. So to summarize aerobic cell respiration, the first step is glycolysis, where glucose is converted to pyruvate. It produces 2 ATP, 2 pyruvate, and 2 NADH. The next step is the transition step and the Krebs cycle, or the TCA cycle. In this cycle, this is where we produce CO2, 2 ATP are created, and we have more NADH and FADH2. And then finally, we have the electron transport chain where 34 ATP are created. So 34 plus 2 plus 2, the total amount of ATP from aerobic cell respiration in bacteria is 38 ATP. And I want you to know the order of the steps. So it's glycolysis, then the transition step plus the Krebs cycle or TCA cycle, then the electron transport chain. And in bacteria or prokaryotes, glycolysis happens in the cytoplasm. The acetyl-CoA transition step and Krebs cycle also happens in the cytoplasm. And then the electron transport chain happens in the cell membrane. In eukaryotes, glycolysis happens in the cytoplasm. The Krebs cycle happens in the mitochondria, and the electron transport chain happens in the mitochondrial membrane. And I want everyone to know the order of the steps, how many ATP are created in each step, and where, for example, if I ask where CO2 is created, I want you to know that it is created in the Krebs cycle. So that's respiration. And in aerobic cell respiration, oxygen is the final electron acceptor. In anaerobic respiration, it's an inorganic molecule, so something that's not O2. So here, let's do a summary last. So aerobic cell respiration uses oxygen as the terminal electron acceptor, and the end product is water. You get about 38 ATP in aerobic cell respiration. Anaerobic respiration is only done by bacteria. So humans, we cannot do anaerobic respiration. In anaerobic respiration, you still have glycolysis, still have the Krebs cycle, and you still have the electron transport chain, but the difference is that the final electron acceptor is not oxygen. It's an inorganic molecule such as nitrate, sulfate, or anything else. This is the reason why bacteria can grow everywhere. The reason why we see bacteria growing everywhere, unlike humans that need certain conditions, is that some bacteria can do anaerobic respiration and can respire different chemicals for energy. Some bacteria can break down nitrate. Some bacteria can even break down iron for energy. And that, again, is called anaerobic respiration. And then the end product is nitrite or hydrogen sulfide, depending on what your inorganic molecule is. And then the ATP yield of anaerobic respiration is always less than 38 ATP. So if you as an organism have the ability to do aerobic respiration and anaerobic respiration and oxygen is present, you would always do aerobic respiration because you get more ATP. Anaerobic respiration varies with the organism and what it's respiring, but it's less than 38 ATP in the end. So if an organism, again, has a choice, you can tell I'm really stressing on this question. It's probably going to be a test question. It will use aerobic respiration if oxygen is present. And if oxygen is not present and it can do anaerobic respiration, it would do anaerobic respiration if an inorganic molecule is present. Now we're going to talk about fermentation. Fermentation, a lot of times people call it anaerobic respiration. It is anaerobic. I wouldn't call it anaerobic respiration. So I would say fermentation is an anaerobic process. So fermentation is a type of metabolism that does not require oxygen. That's why we say it's anaerobic. And so with fermentation, you have glycolysis similar to what we saw with aerobic and anaerobic respiration, but there's no Krebs cycle and no electron transport chain. And if anyone remembers, in glycolysis, you only create ATP. So organisms that do fermentation will only create 2 ATP. And fermentation is all about recycling NAD+, because remember, in respiration, we could take all that NADH we created and send it to the electron transport chain to get energy, whereas here we cannot because there is no Krebs cycle, there is no electron transport chain. So here you're recycling NAD+. And so with fermentation, you only produce 2 ATP total. So it's very energy inefficient, but this is the choice of some organisms. So fermentation is done by various microbes, and your bone cells, if anyone's taken anatomy, if there's no oxygen, like if you're breathing a lot and you run out of oxygen for a little bit, those cells can do fermentation. And fermentation has many different end products. Examples of end products of fermentation, it depends with what you start with. So there's the lactic acid fermentation pathway. So its end product is lactic acid. So some Streptococcus and Lactobacillus species can do lactic acid fermentation. They produce lactic acid and we use it to make food products. Another byproduct of fermentation is ethanol. So ethanol is used to make alcoholic drinks, and then CO2. So when you add yeast is a microbe. When you add it in baking, it produces CO2 as a byproduct of fermentation. So these are all different byproducts of fermentation. So you can create ethanol, CO2, you can get acetic acid, you can get ethanol, you can get gas. Some people use fermentation to try to create biofuels. So many end products are used in our day-to-day life. In lab, students sometimes use Clostridium bacteria, and Clostridium smells terrible. So when you open the tube, it smells really bad. And the reason it does is because of fermentation and it creating gas as a byproduct. So again, fermentation only produces 2 ATP. An example of end products made by fermentation are lactic acid, which we use in yogurt and cheese, ethanol to make alcohol, CO2 used in baking, and many others shown in this table. So we talked about catabolism of glucose, which is a carbohydrate, but you can catabolize you and bacteria microbes can catabolize other molecules to produce ATP. So lipids and proteins can also be used as energy sources to produce ATP. The difference is that if you're using protein and lipid, they enter the pathway in different ways. So for example, proteins are broken down or hydrolyzed to amino acids, and amino acids can enter glycolysis or the Krebs cycle so that you eventually make energy. Lipids can be broken down or hydrolyzed into glycerol and fatty acids, which will enter the cycle back so that you get energy. So they can enter the Krebs cycle to make NADH and FADH2, and then those can go to the electron transport chain. And so you can catabolize many different molecules, macromolecules. One thing I want everyone to know is exoenzymes. So exoenzymes are enzymes that a cell makes and they exit the cell. So some bacteria make exoenzymes. And then what they do is they break down macromolecules so that those macromolecules can be brought back in the cell, the monomers, and used for energy. So examples of exoenzymes are gelatinase and protease. We know they're enzymes because they end in ASE. These are used to digest proteins. Gelatin is a protein to amino acids so that the cell can take the amino acids and throw it in the Krebs cycle and acetyl-CoA transition so that you can get energy. Lipase is an exoenzyme. Lipase is released so that it can break down fats into glycerol and fatty acids, which are then easily brought in the cell. And glycerol can be brought into glycolysis. Fatty acids can be used to make acetyl-CoA. And then amylase and cellulase are carbohydrate exoenzymes. So they're used to break down sugars or carbohydrates to their individual monomers. So know the definition of exoenzymes, and then these are examples of exoenzymes. So lipase breaks down fats, gelatinase and protease break down proteins, amylase and cellulase break down carbohydrates into monosaccharides. So in all the previous lectures, we were talking about catabolism. Catabolism is when you take a molecule such as glucose and you break it down to get energy. Now we're going to briefly talk about anabolism. Anabolism is the opposite. It's a biosynthesis pathway. So it's how do we make carbohydrates, proteins, lipids, and nucleic acids? And in order to make things, you need energy. So with anabolism, you take monomers. So you can take monosaccharides, different monomers of sugars, and you can create a polysaccharide. You can take amino acids and add energy and create a protein. You can take fatty acids and glycerol and create lipids. So photosynthesis creates sugar for us. So that's an example of an anabolism pathway. So keep that in mind. So anabolism is when we take monomers and we build them into their polymers, and it requires energy. So it's an endergonic process. Now, if we want to integrate, usually catabolism and anabolism are integrated. Things are being broken down, which is catabolism, and things are being made, which is anabolism. So metabolic pathways where we see both catabolism and anabolism are called amphibolic pathways. So an example of an amphibolic pathway is the Krebs cycle. In the Krebs cycle, we see catabolic pathways. So things are broken down, and we see anabolic pathways where you are also building different molecules. So if a cell needs energy, it will do catabolism. If a cell needs to make nutrients, it will do anabolism. But the Krebs cycle, we see both examples. We see some components being broken down, which is catabolism, and some being made up, built up, synthesized, which is anabolism. And here is a summary of it all. Now, I want everyone to know that it's a lot more detailed than I explained it, but I want everyone to back away and know the big picture of metabolism. So if we talk about 3 different metabolic pathways, in respiration, there's aerobic respiration and anaerobic respiration. Aerobic respiration uses oxygen as a final electron acceptor here. Anaerobic respiration uses an inorganic molecule that is not oxygen. Aerobic respiration produces 38 ATP. Anaerobic respiration produces less than 38 ATP. In both aerobic and anaerobic respiration, the 3 steps are we have glycolysis, which is step 1, acetyl-CoA, making acetyl-CoA in the Krebs cycle, which is step 2, and then finally the electron transport chain, which is step 3. And then as a summary, glycolysis produces 2 ATP. acetyl-CoA produces CO2 and 2 ATP, and then all the NADH and FADH2 produced go to the electron transport chain. In the electron transport chain, the chemiosmotic gradient ends up creating 34 ATP. Fermentation only has glycolysis. Fermentation is an anaerobic process, meaning there's no oxygen present, and you only get 2 ATP and you get a byproduct. You can get an end product. You can get lactic acid, you can get ethanol, you can get CO2, you can get butanol, you can get so many different end products depending on the organism doing fermentation and what it is fermenting. So fermentation is done by microbes to make us all our yogurt and cheese. And so again, here to summarize it all again, aerobic cell respiration uses an electron transport chain. Its terminal electron acceptor is oxygen. We have the amount of ATP made by everything that is not the electron transport chain, which is called substrate level phosphorylation. If you're watching any other YouTube videos, is 4 ATP, 2 from glycolysis and 2 from the Krebs cycle. Krebs cycle and TCA cycle are the same thing. Oxidative phosphorylation is the same thing as the electron transport chain. So we get 34 ATP. The total ATP we get from aerobic cell respiration is 38 ATP. Anaerobic cell respiration also uses an electron transport chain. The terminal electron acceptor is anything that's not oxygen. It's an inorganic molecule, and you make less than 38 ATP, but more than 2. Fermentation does not use an electron transport chain. It does not use a Krebs cycle. You only have glycolysis, and you end up producing 2 total ATP and then end products such as lactic acid, which is used in cheese and yogurt, ethanol, which is used to make alcohol, CO2, which is used in baking. So much. And so finally, I want to say that organisms can be classified based on the metabolism they use, and we're going to talk all about this later on. So organisms, if they're obligate aerobes, so some microbes, we classify them as obligate aerobes. If you're obligated to do something, it means you have to do it. So an obligate aerobe can only use aerobic cell respiration out of the 3 respirations we talked about. Obligate anaerobes use anaerobic respiration or fermentation. So they will either do anaerobic respiration if an inorganic molecule is present, or fermentation. If they had a choice between the 2, they would do anaerobic respiration if an inorganic molecule is present, because it will always produce more than fermentation. Facultative anaerobes can do all 3 types of metabolism. They can do aerobic respiration, anaerobic respiration, and they can do fermentation. And if given the choice, if oxygen is present, they would do aerobic respiration because they would make 38 ATP. Then if oxygen is done, they will do anaerobic respiration if an inorganic molecule is present, because it will still give them more ATP than fermentation. And if no oxygen is present and no inorganic molecules present, they'll do fermentation, which will give them 2 ATP. So here are the study questions. I know that this is a lot, but I just want you to take it slowly and try to understand the big picture here. So remember, in respiration, the steps are glycolysis. Then we have the transition to make pyruvate to acetyl-CoA and the Krebs cycle, and then the electron transport chain. With fermentation, we just have glycolysis. So just go through, and I put some good YouTube videos. There are a ton of really amazing metabolism YouTube videos.